Hello!
Hi there.
Welcome to Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny, and in a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC, dedicated aiming the public with the tools of biotech.
My name is Faz, and I have a PhD in microbiology. I've mostly worked on bacteria, but I've also worked as a research integrity specialist, and I'm currently working as an editor for a scientific journal.
Every week, we meet to talk about microbiology, and today it's a news week where we do an overview of some of the coolest microbiology papers that we've seen this week.
And at the end of the month, we'll be selecting a paper to do a deep dive into, so we've got a poll running on our Twitter, so I've linked to it in our comments, so if you want to vote in it, vote for any of the papers we've shortlisted, then please do, and whichever gets the most votes, we'll be covering at the end of the month.
Yep. You can follow along with the papers that we discuss on either week, news or deep dive, in our shared Zotero library linked in the doobly-doo below.
And we want to hear from you, so please use the comments or tweet us at microtwjc. And boy, do we have a show for you. First, we have the structure of the Omicron spike. Then we have a look at what we can learn about COVID in the heart from hamsters. We learn about a new component of the human blood that we produce to fight SARS-CoV-2. Then we have a look at long COVID and also the impact of vaccination on the menstrual cycle. But not only that, we'll be talking about how hairy balls can be the next big antibiotic, how kombucha might be used to filter water, and how viruses that kill bacteria swap genes, and finally, a molecular valve that will allow bioengineers to control RNA transcription.
Yeah, that's a long list. And as always, we have a mix of SARS-CoV-2 and other topics. And we're starting off with the structural basis of SARS-CoV-2 Omicron evasion and receptor engagement. A really fast structural biology paper on Omicron. I feel like this is the power of cryo-electron microscopy. Is you can get structural information quite quickly. This was the same technique that was used to initially find the structure of spike way back when. Do people remember that? Two years ago almost?
What happened 2 years ago? So, I mean, who can remember anything? It's all just been 1 year, I feel like 1 year since 2020. But yeah, I mean, with Omicron, like, we've had some like kind of simulations and some predictions about what the structure might look like based on computer, but this paper, they actually look at how it actually is. So using X-ray crystallography, which is electricity, but also cryo-EM, which we're quite interested in, where you freeze like these like proteins and then take a very thin sliver of them and use some very like clever like mathematics and laser light to figure out what the shape of these molecules are. And the great thing about it is also, because you're looking at lots of them at the same time in different conformations, you also get to see like how its shape can change and alter, and kind of like how it's a bit more wobbly than you'd kind of expect.
Yeah.
But
what are the takeaways? I feel like one of the takeaways that it seems like they talked a lot about is there's a lot of mutations, or there a subset of mutations in the interface between the monomers of the SARS-CoV-2 spike. So the spike is a trimer, if folks remember. The Omicron variant does have mutations that lie in the interface between the monomer units. But yeah, that's kind of like, okay, so there's some difference in the way that these get constructed, I guess, is the takeaway there.
Yeah, I think a lot of this is mostly like confirming stuff that people had already suspected based on the sequencing. So there's a lot of stuff on like where the antibodies are supposed to bind, and also and how it's like, because the spike protein has an open and closed conformation, so like how that is changed slightly in Omicron. But mostly, like, I feel like this is a good like reference and source for people if they're interested in the structure for Omicron and looking at the differences, because it kind of like pulls in everything we know together and has some confirmation. Because before, like with simulations,
Totally.
Guesswork.
Right. Yeah. So like, for example, like we've seen the in cell culture like data that says that Omicron, and even in animal models, we've seen that like, oh, Omicron maybe binds differently to the ACE2 receptor, right? And like everyone says, like that's because of the mutations in the receptor binding domain. Well, of course, right? That makes logical sense. But here in this paper, we can see it, right? Like, well, now they have an image, right? I think in their last figure, they point to, oh, okay, well, here is an actual structure basis, right? That has the Omicron spike with the receptor with the ACE2, and you can see like there are some interactions that don't exist anymore, and there are some new interactions that it has.
Yeah, completely. So it's, I mean, because I think this week I've been a bit like slow on finding Omicron stories because there's a lot of stuff on Twitter, but not much in actual like published form that stuck out to me. And also I realized that for the past few weeks I've mostly been doing preprints rather than actual papers. So I'm trying to
Yeah.
break that habit a little bit. Um,
Yeah. See with some peer-reviewed stuff maybe that folks might be interested. I guess, but it also shows in some way. One of the things that this kind of sheds light on as well is just like that cycle, right? That it sort of takes some time for peer-reviewed things to come out, and there's a lot more Omicron in the preprint world than there is inside of the peer-reviewed world.
Yeah, completely. So I think we can move on from this paper because this is fairly straightforward to another paper, which is the golden Syrian hamster as a model to study cardiovascular complications associated with SARS-CoV-2 infection.
Yeah, so this model, we've heard about the golden hamster model before. Hamsters have an ACE2 that's more similar to humans than it is to their rodent brethren. And that makes them great for an infectious model because they do get sick when you give them SARS-CoV-2. And this model, they're still using the intranasal infection route. But instead of zooming in on the lungs, they are actually looking at the spleen, the lungs, and the heart. Looking for pathology, and this is a characterization of a model, and when they characterize a model, it's important to see it in different circumstances where you put more PFU, right, more infectious disease. You want to see histology of all lungs, like what does this infection look like on the animals as a whole, and then you can potentially position the model to be used to answer a more specific question about how that disease process works. So then they go into like some metabolic sort of insights into cardiovascular disease.
Yeah, yeah. I mean, that's a really interesting thing about this model because we've seen it before. I mean, I think we've seen in hamsters how SARS-CoV-2 can spread to the brain, and this is like so. I think. And now it's not so much that they're doing anything different. They're just looking in different places because it is quite a good. And I know that recently there has been a lot of like to do about hamsters because I think in Hong Kong there's been a hamster cull because I think this week they've had the first like hamster to human infection.
I see.
Yeah, I mean we've seen stuff like that before. We've seen ferret to human infections in the Netherlands. I think there's been. I mean, correct me if I'm wrong. I'm sure there's been like a domestic cat like infected by humans and vice versa.
I'm not sure about the vice versa, but I think in one direction.
Yeah,
I know
think
that
maybe
zoo animals, right? There in the beginning we covered a bunch of news things. Zoo animals were getting infected.
Yeah, so yeah, SARS-CoV-2 can go into and in the case of hamsters, it can tell us a bit about what's happening. So in this case, they look
because
at
they're.
Yeah.
I just want to say because their disease, because they have a certain type of disease, then we can say that might be similar to ours, right? And that's the logical bridge. And then, they're mammals as well, so some of the signaling pathways might be conserved. I like figure 2 actually that we're looking at right now. Probably we knew this before, but it just reminded me that for figure E, actually, right? There's at 2 days post-infection, like there's a huge jump in like all these different cytokines, but then they actually go down after a period of time, but some remain high. And just like that, the nature of how the immune response, it's like interesting to always see that again. Again, I'm not really sure like how I would use that information in my day-to-day life, but like if I was a disease researcher, right, like this would be very useful information for me.
It's interesting because you can never get this thing out of humans as well because for the humans you're never there at the point where they get infected and so you can't find the peak, or if you do find the peaks it'll be in all sorts of different places because people don't know when they get infected. There's a lot of variants where they don't necessarily before time for symptoms to appear, so with hamsters you get that kind of perfect like kind of encapsulation of what the immune response is, and then from that you can look at symptoms and biomarkers. So I think what they found here was that there's like a biomarker that's shared between hamsters and humans,
Yes.
a lipid that they characterize.
Yeah.
They also see some basic stuff like, so histology showing that the heart becomes thicker and heavier, and a bit more inflamed. Same as in humans. They also look at the metabolome,
Yes.
and they find that there is let me see. I wrote this
Oh, you
down.
wrote down what it was. It's like a
N-acetyl neuraminate.
so there's lots of different types of fatty acid right? There's lots of lipids in the world, not just, the if people did like a high school biology or whatever, right? Like you know that these lipids like form our cell membranes, but there's a lot of variance depending. Those lipids can be modified in different ways, and yeah. So this metabolic approach is essentially taking blood and putting it through a mass spec so they can find the identities of all these different molecules. It really is, if we've seen before genomics information, right, where you get all this, right, all the genes that are being expressed, transcriptomics, right, all the genes that are being turned on or off in a scenario. This is taking that that big data approach, but applying it to the molecules that exist, and you're sort of creating this huge inventory of every molecule that you have in a specific sample.
Yeah. So yeah, this is pretty interesting. And it just
Look at
kind
that
of
hamster figure.
yeah.
How cute.
I mean, I do love it when scientists draw animals. It can be quite fun. Yeah.
Um,
What a happy hamster with
what a
a
happy.
spleen and yeah. Uh, let's take it in. How about we move on to the next paper, which
Yeah,
is
absolutely.
yeah. Circulating ACE2-expressing extracellular vesicles block broad strains of SARS-CoV-2. Now,
Yeah,
this,
this is super weird.
yeah,
I didn't
yeah,
because okay, so first, this is the first I've ever heard of extracellular vesicles in humans. We've talked about them on the show in respect to bacteria, right? And
yeah,
yeah, that like some bacteria have ways of like blebbing, right, the surface of their membranes out. Those things go float around. Then we've also kind of talked about in the context of people make vesicles that have ACE2 on it, and it kind of soaks up SARS-CoV-2. Or like it was maybe they make vesicles. They just made like ACE2 like nanoparticles, right? And they have like soaking up all the SARS-CoV-2. But this picks up a thread of extracellular vesicles in humans. So I guess sort of a new thing people can have discovered, right? Like that we have extracellular vesicles floating around in our plasma, and if they have ACE2 on them, you would expect that they kind of have that same mechanism of those ACE2 decoys that are right as a therapeutic. And yeah, here they're trying to narrow down the function or show that it's not really, we don't know what their exact function is, right, in the human body, but show that they do have an effect within SARS-CoV-2 infection.
yeah. Super weird. The idea that like we as humans kind of are producing extracellular vesicles, and that in SARS-CoV-2 infection, not only do we produce them, but the amount that have ACE2 are like increased. That is like, I have so many questions. Like what? So what is happening? Where in the body are these extracellular vesicles being created?
Yeah, what
What
cell
is?
type do they come off of? What genes
Yeah,
control
I
that
mean,
process?
yeah. So like I kind of like did some digging to try and figure this out.
Oh, okay.
Like yeah, I didn't really get too. So like it's apparently extracellular vesicles are one of those things where they're not easy to detect in the blood, which is why we often ignore them. I think people have been using them as like kind of a source for biomarkers of like cancer usually.
Mm. Yeah,
But
because
also
they're
like
membranes, right? I think maybe that's like a nice
yeah,
part of it. It gives you, because when we think about proteins in the blood, right? Proteins in the blood, like they might be dissolved, right? Biomarkers get dissolved in there, but if these if these are coming off of certain cell types, then those cell types have lots of cell membrane proteins, right? That kind of expands the list of things that you might be able to detect. And yeah, interesting to hear it come from the biomarker world.
Yeah, and so like with respect to SARS-CoV-2, what I've read so far is that there are certain like aspects of SARS-CoV-2. I think that so I've read a few papers, so I skimmed a few papers. I didn't
Yeah.
like read it extensively, but they seem to indicate that SARS-CoV-2 could force cells to make extracellular vesicles in order to evade the immune system.
Okay, sure.
And
I mean,
and so
we know that SARS-CoV-2 does have the ability, or I mean, a lot of viruses in the positive stranded RNA world, right, have this, right? They make these membrane-bound things inside of the cells in order to have replication. What do you call them? Replication sites.
oh yeah, organelles,
Yeah, replication
replication
organelles.
organelles.
Yeah. So that they have some, right? They have interfaces to that aspect of cellular biology. Yeah, it could be an extension, right, of that. Like maybe it's like a funny immune thing that happens
Yeah,
because they're messing around with that. Yeah, we don't really know which way it's driven. I mean, there's always a challenge, right, when we talk about a virus, right, interacting with the cell. What part of that interaction is for the virus's sake, and what part of that interaction is like tripwires, right, that let the cell be like, alert, something's going on.
yeah, because both the virus and the cell want the immune system to not be too high, because
Hmm.
of course the immune system is bad for both if it goes into overdrive.
Right,
And
right.
but part of me was thinking that like there is like it does seem like a very neat like addition to the immune response, where if your cell gets like attacked by a virus and it starts blebbing off like extracellular vesicles that have all the surface attachment proteins that the virus used, then that could be almost like another layer to the immune response, which
Hmm.
should be an interesting like kind of hypothesis to work off of. Um, but I mean, yeah, there's a lot of like people saying different things about perhaps like it also being beneficial for the virus because it modulates the immune system. So it's hard for, but in this
Yeah,
study they
yeah. It's hard to make that. It's always like to take this observation and then try to link it back to its exact function in the biology of things. But what we can
yeah
say in this paper is that
yeah
they make well, yeah, they do it two ways. They have a cell line that makes these extracellular vesicles, and then they use that to show that it prevents infection in a cell culture model. I think they also then take those same ones and they put it in a mouse model, or maybe they switch over and they find that the
yeah
ones in humans are also useful to suppress infection in a cell culture model, and then inside of a animal model as well.
yeah I think. And that's just a very interesting perspective because I do remember like early on the outbreak, lots of people were experimenting with ACE2 like what is it, dummy or
Yeah,
like kind of
decoys.
bait
Yeah,
decoys? Yes.
we have a deep dive on ACE2 decoys. I feel like we always go back to it because it's actually a really interesting paper to think about in the context of just what mediates binding between the spike protein and ACE2. But yeah, in this instance, it's like, yeah, just the fact that you have something, a scaffolding, right? These essentially that's what these extracellular vesicles are, a scaffolding for ACE2 that our body can make and have them floating around, will absorb up the pieces of SARS-CoV-2 and prevent them from gaining entry in a cell. Because if a SARS-CoV-2 fuses with a vesicle, it's not going to have the same machinery that it needs inside of there. It's essentially an aborted infection.
Yeah.
But
So I mean,
oh, sorry, but I do want to just
no.
hold this thought that without knowing how they're made, like the big experiment that would really wow me is like if people knew how they were made and you could have a transgenic mouse that didn't make them, right, or something like that, and then like show that like when you knock that out, then the infection is worse. Like I think that's like a really interesting like thing that would just change the way I think about infection.
Yeah, completely. I mean, I think
Mhm.
that this is still extracellular vesicles are still a very like new science. I think, and
Right.
I think like that question. I think a lot of people are asking,
Yeah.
and it's gonna be
Yeah.
quite interesting because yeah, I didn't. I mean, admittedly, I only took about like maybe half an hour of trying to find out what the answer to that was, but I couldn't even find like any.
Yeah, I didn't
So
see anything in my cursory glance either. And I do want to throw one more interesting thought out is that for anyone who's looked at anyone who's looked at cell flow cytometry right before, like that's the tool that they use to find these extracellular vesicles. Like normally you just say like that stuff's junk, right? Like it's not big enough to have what you want. It's staining with the things. It might be staining with your preferred cellular marker, but like it's too small. So it's like you just like ignore it. But I just like this is like one of those wonderful things about science. Is that like, but then actually that data was in front of us the whole time, right? People could go back to those things and be like, oh gosh, like, is that what this part of the graph is? Like, we just always ignored it, but actually it's extracellular vesicles. Yeah.
Yeah, I mean, that's a really good point. Like, because finding one man's junk is another man's treasure in that kind of terms. Yeah.
You don't know what it is until you you're paying attention to it, right? Like the world's such a big place. In biology, we have all these tools, right? It's not like direct looking where it becomes so evident that like, oh, that's a strange thing I should pay attention to, right? It's like a blip, and then until functions get assigned to it, scientists aren't paying attention. So it is important to I don't know, keep an open mind sometimes when we're looking at data. And for people who know how to look at a specific graph, right, like that is a very rich source of knowledge, right, to be able to look at a graph and say, this is what this part of it, even we're not paying attention to it and we translated it into a bar graph later, that original data collection graph like has a lot of richness in it that might have yet to be appreciated in many systems.
Yeah. Cool. I think that brings us to our long COVID paper, I think.
Oh, yes.
Multiple early factors anticipate post-acute COVID-19 sequelae.
Mm-hmm. So this is an interesting paper because, right, it's quite difficult to collect robust data sets to study something like long COVID. So here they're calling it, right, post-acute SARS Sequelae, what? Post-acute COVID sequelae, right? PASC. But essentially, this is a stand-in, maybe a more technical term for long COVID symptoms that persist after your acute infection. And the reason why that's difficult to study is because you would want information from the acute infection as well as afterwards. And then there's also this very difficult part of how do you collect the symptoms right afterwards? There's going to be reporting biases and lots of different things. So in this paper, they have one data set of people that they have 3 different blood draws from their acute infection, right? From their and I think maybe during the infection, something like that, and then like sometime after when they're ostensibly experiencing these post-acute symptoms. And then they also have another data set of people that were just like directly after, that they only have the post-acute symptoms. And the idea behind their paper is they want to sort of figure out some associations inside of they have all sorts of, right? They have blood samples, so they can do all sorts of, they don't metabolomics, they do like cell-based assays. Yeah, you're gonna have to like scroll all the way to the end to find a good graph. I think it's like 46, page 46.
Okay, let's go page 46, which is that one. Okay.
Yeah. If you zoom in at the top of this one, you can see sort of the selection of different things that they're looking at. They look at metabolomics, they look at neutralizing antibodies, they look at SARS-CoV-2 in the blood, the RNA in the blood. They look at they look at single-celled sequencing of the immune cells. They're really just getting the widest amount of data that they can from the blood side, and then they're trying to find associations in those data sets to the existence of these symptoms that are going past the acute infection. And in doing so, they're able to associate some pre-existing conditions with the likelihood, right, of seeing those post-acute symptoms, and then they also associate like the presence of RNA inside of the blood, the presence of Epstein-Barr virus in the blood. So again,
Yeah.
these are just like correlations type things, right? There's no way for them to know the directionality of some of this. And I think they do a fourth thing, but I don't quite remember. So they find a bit, they're fishing, right? They're fishing for associations, essentially.
Yeah, it's like an essential part of like the first stages of trying to study something. But at this
Yes.
point, there is always have to be that wariness that like you are going to get a few spurious associations. And at this point, we're just kind of formulating the question because
Mm-hmm.
because I think one of the interesting things they do in this paper is usually we think of long COVID as just one disease, but actually they've broken it down into sub diseases with different symptoms. And
Yeah.
I'd like to see that developed a lot more because I kind of feel like there's a lot of complicated things happening uh, and
Yeah.
and I feel like a simplified viewpoint may not be helpful. I think there is going to be a lot of diversity, and they recognize that there is a lot of diversity in how people have long COVID, and I think that's a
Mm-hmm.
really important thing to pay attention because if you try to
Mm-hmm.
like find the average person who has long COVID, you're going to like erase a lot of people's personal experiences, and
Right.
I think
Well,
that
also like you also might not be able to find an answer, right? If you're trying to
yeah,
find in the average person, right, with some post-acute sequelae, like if the mechanisms of creating those symptoms are different between right those individuals, then an average of them we may never be able to trace back and learn about the mechanism of how it all developed. So I think that that's probably a big part of why they decide to split things is because they want to encourage researchers to find mechanisms for this stuff, and the most logical way to do that is to try to try to split out. Oh yeah, Minref asked why are they looking for RNA in the blood rather than the tissue. It's because they have blood, right?
yeah.
Like
Why are
this
you
is
looking
the
at yeah?
yeah, getting trapped in the data set that you have, right? It's so these people are, right? This is not autopsy stuff. This is a
Mm.
longitudinal study where they enrolled a section. And so, yeah, it would be really cool to see. I mean, especially because later on in this paper, they start developing a hypothesis about gastrointestinal
Mm.
post-acute sequelae, and they start linking it to changes in immune cells and the way those immune cells are developing. Well, a big part
Ooh.
of immune cell development in the gut is like tissue specific. And so like you wouldn't see some of the potential markers unless you had a tissue biopsy, but they didn't know that until they did this analysis. So like that cohort is gone now, right? Like you can't go back and, well, I mean, it's very difficult to go back and ask these hundreds of individuals to now like have an endoscopy. Well, I think actually, first of all, that's not done because that's a risky like
Yeah.
operation just for research, right? So like these types of materials are not like freely available to do research on. Yeah.
Yeah, I know that bit we mentioned about the gut. I mean, that reminded me of the paper of the evolution of immunity, which was one that we go back to quite a lot, where they had this theory that potentially SARS-CoV-2 could be still in the gut, and in
Hmm.
itself could be almost like a different manifestation. So if you were to say
Yep.
that all long COVID is due to that, you might miss out a lot of other things that are related to the factors they're looking at here. So
Yeah.
it's complicated.
Yeah, but
And
so
yeah,
like maybe let's just go to a figure. Like I think it's page 49.
page
This just gets at
49.
one of the um, this gets at like
Right.
just from the blood they're able to yeah, this one. So just from the blood they can pick out immune cells, CD8 immune
Mm-hmm.
cells, right? And they can look at them, right? Compare them between the acute phase and the convalescent phase and say that they're behaving differently, right, in people that come out with post-acute sequelae versus not. And I think here they were able to uh, just pick out the folks with symptoms. I think they focus on the gastrointestinal ones for this, right? So to build this association, they focus on one set of symptoms, and then by focusing on that set of symptoms, they're able to pull out pre-existing difference inside of the acute samples, right? That precede that symptom. So, right, it's just a correlation at this moment, but it's that like tasty little breadcrumb that like someone can pick up and say, so now how do we design experiments to, if this is the hypothesis then, right? Like it creates a hypothesis. The hypothesis
Yeah.
is, right, something is going on in the CD8 T cells, right, in your acute disease phase that means that results in, right, the post-acute sequelae. So now you'd have to go back and think, how do you design an experiment to get at this, right? You're not going to be able to do it in humans probably, right? You're going to have to figure out an animal model or like a reconstituted biochemical immune system, right? That you can do this. Oh, maybe an organoid, right? Maybe you can do this in an organoid culture. But that's what this study is bringing to the table. Is it has the difficult to get human cells. It did like a crazy data collection, right? Crazy data collection sort of effort, and then they've done these associations to try to point people to potentially the most fruitful research lines to follow up on.
Yeah, so this is like, I mean, hypothesis generating research is a really important part of science, and it helps you circle around the right questions.
Yes.
So yeah, yeah, it's quite nice how comprehensive this data set is, because it means you can go back to it and then look back and you get more context on things. So I think this
Yeah,
is quite
totally.
interesting.
Yeah, I thought
But
it was
yeah,
definitely good news for people who are concerned about like how are we going to deal with these post-acute sequelae. Researchers are beginning to build some picture of it, but we're not
yeah,
quite sure what it means yet.
yeah, it's news. I wouldn't say it's good. We're getting to a point where we're gonna start to try and ask the right questions about the disease. I'm not sure it's like indicative that we're gonna be able to do anything about it at the moment. I don't think it's that hopeful. I think it's just an important stage.
Yeah, okay, sure. Not that hopeful. I mean, like to me, just knowing about a thing is hope. Like even if you find out that you can't do anything about it, I don't know. Like that's something that science can provide this.
Yeah, I mean that's great.
Maybe
Don't look up. It's
that's
just
my brand of hopefulness.
yeah. But cool. Let's bring on to the next paper, which
Yeah.
is called Association Between Menstrual Cycle Length and Coronavirus Disease 2019 Vaccination, a
Hmm.
US cohort.
Yeah, so this is an interesting one. The strength of this data set is that there's a cohort of people that are using an app to track their menstrual cycle, and
Right.
they were using that sort of before before COVID, right? Like this is just like part of their daily practice or whatever, and so they're able to consent. So I mean, it's always very complicated, but they had a
Mm.
pre-existing consent, right, to use this data, right, in research, a de-identified set of this data in research, and so they can go and they can look at this now to try to cut through the idea that with self-reporting there could be like right, it's not so precise. How do you know like exactly when if you're just reporting after the fact when you got your vaccine and when you notice changes in the menstrual timing. So that's what's great about this data set is that it's sort of, even though the analysis is retrospective, the collection of the data was prospective. Not for this purpose, but it was.
Yeah. So that's quite a strong thing. I mean, again, I feel that we do have a kind of weakness in this podcast because none of us have experienced this kind of thing that is a daily, well, not daily, but
Definitely.
a
This kind of
regular
thing.
part of.
Yeah.
So
Yeah.
yeah.
We're not menstruating. So like again, that's also why in some ways I hadn't been following the news on this until someone mentioned it to me. I was like, "Oh, I should go hunting for a
Yeah.
paper, right, in this realm." Because I think it's becoming more of a concern, right? As you get more anecdotal reports, right, of
Mm.
people's experiences and the shift that they're experiencing, then people have to ask, like, well, how can we study this sort of en masse? How can we understand more? And then this is an attempt at that.
Yeah, I think that this is yeah, and what it does find is it's
Finds like
here there's
a 1 day difference
yeah,
in
it's
this data
so
set.
yeah. So on average, it's like less than a day, but that just means for most people in this data set, there isn't much of a difference after the
Yeah,
vaccination. But there are some subset that do feel some kind of a difference, and
very much
uh,
so. And another limitation that they explain in this data set is that they're looking at people who use this app who have a specific like profile, right? So, and then they also, I think they also excluded in this analysis, they also excluded people that don't have a really regular cycle, a very regular menstrual cycle. So, right, like there is variation within individuals, right? And not everyone falls into this one category. So the conclusions that they're taking only apply to that limited set. And so again, like we don't know. It's still very much like an unknown effect, right, of the vaccine on menstrual timing. But at the same time, from the snippet of information that we have, they make the conclusion that there's no clinical significance. So still get your vaccine, and then it's about like weighing risks again, right? Like this is like, then they want to weigh risks. They say, okay, well, think about if you did get SARS-CoV-2, there is a known effect, right, on sort of blood. Yeah. So they want the end result of this paper is trying to encourage folks, I think, to not worry about getting your vaccine in relation to the menstrual cycle, but it also exposes some threads where you can say, but there might be an effect, right?
Yeah, I think what'd be interesting is if they like looked at other kind of factors as well to because I mean, let's say like people who have had flu vaccinations or other kind of lifestyle things, but of course they didn't have that because I think with this app they actually added in some functionality for people to mention that they had the SARS-CoV-2 vaccination, right?
Yes.
So
yeah, so
that was
they don't have it. They don't have the ability, right, to sort of do the counterpoint, right, of another vaccine to see if that difference is within, right, the statistical bounds of just like getting a vaccine and just this is how it works. Because there is, they mentioned this inside of the discussion of the paper, right, that it seems like from first principles, you would actually think it matters when you get your vaccine, right? Because that moment is when your immune system bumps, right? But the menstrual cycle like has a specific sequence of events that's happening. So like the idea is essentially you're stimulating an immune response at a very specific moment, and maybe that's what's causing this small change. And again, they say for this cohort that they study that there's no clinical significance to that change, right? That people, that's what, right? People have variance in that range anyways.
Because I think that this is just an important thing to keep in mind because the impact of I feel like there is a lack of like kind of study in the impact of the menstrual cycle on like in infections on anything, because it just seem like, because I mean I remember like back when I was researching, I was working with like female mice and had to keep into account how the cycles interact with infection, because there are certain points in an infection where the disease would go in a different direction, and so for a lot of research we kind of see that kind of elided away, and we need that's why I'd be interested in seeing what like other vaccination effects might be, just so that we're aware
to have a baseline understanding. And again, this may just be our ignorance in this area that knowledge might be out there, but like you're saying like to have a baseline understanding of like how a vaccine, right, an immune stimulation would affect the menstrual cycle.
Yeah, because I mean we went through like a whole bunch of like vaccine papers and nothing like this was mentioned, and I think and when it comes to pregnancy, mostly they go, we don't know yet, and then they wait until other reasons. And I feel like maybe like if we could get that information all together in one go, that'd be really helpful for people.
Well, I mean, to me it's like a blind spot in research, right? Like people are, I think, aware, right, that like and again, it's kind of the same point that I was saying before with like looking at the graphs, right? Like you only know what you collect, right? And because clinical trials, like they take money and stuff, and people just want their drug out there in the world, and I think we have said this before, right? Like they restrict the type of information that is being studied in those things, right? They try to laser focus on what would show people, right, something positive. And so some of these studies, right, like get done later. It almost doesn't mean they're super important to have. And it's just like the incentive structure, right, isn't there to get them up front. Because ideally, we would have this information up front before we started. But like, I don't know, that's not the world that we live in, unfortunately.
Yeah, that's a depressing
Yeah.
note. So let's move on
Okay,
to
yeah, let's move on.
let's
Let's move on.
end our talk about SARS-CoV-2, and now we're going to move on to like non-SARS-CoV-2 stories. So our first one
Yes.
is a paper titled "Hydrophilic nanoparticles that kill bacteria while sparing mammalian cells reveal antibiotic role of nanostructures."
Yes.
This is a really fascinating paper for me because usually when you have an
And
antibiotic, it's interfering with some kind of chemical reaction in bacteria. So like bacteria have cell walls, we don't, so therefore we can target that part of them and have them and poison them, and it won't affect
mhm.
us. Whereas this is seems to be a lot more physical. Like they've created these nanoparticles that are covered in these polymers. So these things called like nanobrushes, which are basically like hair. So these are like hairy
Yes.
balls that are made of like structures that are actually chemically inert. They don't attack bacteria. The chemicals themselves don't attack bacteria. Don't
Yeah, they don't do chemistry in the sense of like chemical reactions, but
right.
they have physical properties that are unique, and those physical properties may interact uniquely with the different physical properties of our respective cell membranes, right? Mammalian cells versus bacterial cell membrane.
Yeah. So this is literally like having a. So I mean polymer brushes have been around for a fair bit of time as coatings on certain
Mhm.
like paints where they want to avoid like algae or other bacteria growing. They disrupt the
Yeah.
membranes somehow. Um,
Yeah. Oh, let's go to figure one so people can at least stare
yeah,
at some TEM images and yeah.
yeah. So you got these.
Yeah, I just want to say it makes me think of burrs,
Mm,
right? That seed dispersal mechanism that some plants have, where you like, if you're a furry animal and your surface rubs against one of them, they're designed so that they'll attach to those furs, and then that's a physical, right? That's a physical interaction. And similarly, if I take my skin and I try to get a burr attached to it, like it won't. And so like that's maybe a nice analogy to think about how these nanoballs, right, interact with a bacterial surface versus a mammalian surface.
yeah, and the interesting thing is that the initial when they're looking at these, they're looking at them as like a drug delivery mechanism because what
Mm,
these like polymer balls they have like special like kind of properties. That means they can release drugs at certain times, but like the hair can lie
yes,
flat
yes.
and then they can stick out. And what this
Hmm.
paper, and so they had added antibiotics, and then some people asked, well, what if we just took out the antibiotics and saw what happened? And they found like things like graphene nanosheets can like cut through the cell membranes of E. coli, and they can do all sorts of like membrane attacks. And so they've got these nanoparticle pinched polymer brushes, which like they bring to mind. I don't know if you had those chewable toothbrushes that they sell at rest stops.
Chewable. Oh yeah, yeah, I know. It's
The
it's considered like an old form of toothbrush, right? It's a branch, and you like make the fibers come out by chewing it, and then you brush with that.
Oh, that's a different thing.
Oh, that's not what you're talking about.
These are
Okay.
like
What are you
kind of
saying?
like you take the brush part of a toothbrush, wrap it around stuff, and just have that, and you pop that in your mouth with some toothpaste, and you chew on it, and it like brushes your
I
mouth out. It
see.
is
I
the
see.
most
I see.
horrible thing ever. Don't only if you have no other choice.
Yeah, like chewing a sponge. But you're not supposed to like actually chew it. Like it's just the action of chewing is brushing.
Yeah, um, but yeah, so
Just
they do
chew gum.
some
Right? Sorry.
yeah. Oh gosh, yeah.
Okay,
Yeah,
but that's what we should conceptualize this on the nanoscale, then being released into a growing set of cells, and those brushes they begin to aggregate on the surface of the bacteria. I think they have
yeah,
an image of that too a
yeah.
little bit later on.
Yeah, they basically like have they're testing multiple different brushes of different sizes to see which ones are perfect, and they think
Hmm.
they find like under about 50 micrometers is the size where they're active against bacteria. Anything larger than that doesn't quite work,
Uh-huh.
and it affects most bacteria about the same way. And their first theory was okay, well maybe it's because they're going through the cell walls because bacteria have these cell walls that are permeable and they filter out molecules that are larger than 50 micrometers. So but then they
Hmm.
tested it against gram positive and gram negative, and they found the exact same size limit, which is interesting because gram-negative bacteria, they've got a cell wall, but that's covered in a membrane. So if that theory was right, then somehow these brushes would have to jump through the membrane somehow. So they went back to the drawing board and went biophysical, where they basically started looking at it. It all came down to curvature, I think. Where like
that makes sense. I mean, they're smaller, right, than us, so like they have more curvature than a mammalian cell would.
Yeah. So having brushes that are small and like curved means that they can harm the bacteria without harming mammalian cells, which is just.
Oh, I see. So you're talking about curvature on the brush side is what drives. Yeah.
So like if you make a sphere smaller, technically I think it's more curved. Bigger, then there's more flat space. And if you get as big as the Earth, it's completely flat. And
right.
In
a local environment. So then here they have some TEM and SEM images of just bacteria getting covered in these tiny little guys.
Yep.
Yeah. Strange. Like definitely a weird thing. Yeah. I mean, I guess I'm a little bit. This is like true for. I mean, even when we talk about like really cool drugs and stuff, like, but maybe more because like we're talking about like these physical tiny balls. Like I'm primed to think about like, but what about like what happens to afterwards? Like where do these balls go?
Oh yeah. So
right? Like what happens when these balls get released into the environment and everyone starts using? Like
yeah, completely.
But it's true for every drug as well. Like I feel like only because like I see them, I'm just like my mind's like always about microplastics. I'm just like, oh my god, what's going on here? But like this is also true for every drug that we talk about. Just to like communicate to the audience, right? That drugs too, like you have to wonder like where do they go after they pass through your body?
I mean, here they only test on cell culture, so we don't even know where they go to in the body and how.
Oh gosh, yeah.
So like this is an interesting like thing, just thinking in terms of like how it works. I mean, here it shows that kind of these like kind of hairy hairs like go into the cells and create these pores. But the question is, what the pharmacokinetics of that? What happens when you take these?
Yeah.
They kill the bacteria, and then do they get stuck in the kidneys? Would they end up in the liver? We don't know, and that's a problem for another day. I think this is more like getting to the theory of how they work, and maybe like looking to the future of like other things that can do the same thing without being as.
Yeah, because actually that's a good point. Like thinking about the theory on how they work, it's just another way that maybe we haven't thought about trying to disrupt bacteria, right? And like at the level of engineering, we have the ability now to make these tiny structures. So can we think about like just like a physical engineering problem, right? Of how do we make something that goes in our blood and then collects itself and like gets thrown out, but it has this mechanism of killing cells.
Yeah, I mean, if we can understand this, maybe there's a potential to create a biodegradable version, or
right.
So I think that's quite. But yeah, I think the thing that really attracted me to this was that it is like completely unlike any other antibiotic I've seen. It's entirely physical. It's a spiked ball that bashes bacteria and somehow doesn't affect mammalian cells. It's just.
That is very strange. Yeah, a good find.
Next one.
Living filtration membranes demonstrate anti-Biofouling properties. And so this is sort of a materials paper thinking about the cellulose layer that forms on top of kombucha. I think people often call that a SCOBY. And yeah, so that is made of cellulose and it's full of bacteria that build that cellulose layer. It, I guess, has structural similarities to what water filtration mechanisms that are essentially just pieces of plastic that have tiny holes inside of them. Actually, I think they use cellulose as well. So like, right, like in industrial process, they just build things that have like a little bit of cellulose. And they test these in the ability to filter water. So they put them in a little test cell where they apply pressure to one side. They use water from many different sources, different reservoirs, water treatment reservoirs, and they just look at the properties of how well it filters stuff and how long does it take until it gets jammed up. And they find that the kombucha scobies are quite good, actually, at filtering over a wide range. So they're comparable in the sense of their filtering ability, but they also don't get clogged as easy. is
Yeah.
what is their finding? Mm-hmm.
Yeah, so the thing they're looking at is biofouling, the ability for, because it happens in a lot of systems where you get bacteria just grow everywhere and they can
Mm-hmm.
clog up pipes. But since kombucha, it's a living like colony that produces like acetic acid and other kind of things that are antibacterial. So the
Mm-hmm.
theory is that because it's already a living like membrane, that it can like discourage bacteria from growing.
Yeah. Yeah. So like this is just showing that it works just as well
Mm.
as a regular membrane. But if you go to figure 3,
Figure three.
yeah,
Yep.
it's like on the figure 3. Then this is where they look at those membranes, right? The living filter membrane, the kombucha SCOBY. They look at like,
Yeah.
oh, how many cells can they see stuck to the surface of this thing, right? And it's a lot lower than just like clean cellulose. So yeah, just a fascinating idea, right? Of course, like this doesn't address anything about like how would these be manufactured or implemented or all this sort of stuff. But it's that nucleus, it's that idea that we should be maybe thinking about this material as being good for filters, having properties that are desirable.
Yeah, I had one question. I didn't like get the chance, but will the water taste like kombucha if I like filter it through this, or
Yeah,
am I
I
willing to make
mean,
that sacrifice?
I mean, the process of kombucha is like a fermentation, right? That takes like
Mm,
they're not going to be able to ferment as fast as water is flowing through them. So I
right.
think
Okay.
I think we're in the clear.
Okay. Phew.
But like imagine and this is like crazy sci-fi stuff, but like like, I think this is people's hope for synthetic biology sometimes, right? You then embed. This is actually related to the microbial ink, right? Like paper that we have up for vote, right? Then you embed something that does a function inside of this membrane, and so now like you're processing. Maybe it's like degrading pharmaceuticals, right? Like, in these water filters, you have something that degrades pharmaceuticals, and then you do it in such a way that you're getting extra function from the membrane.
Yeah, or you put malted barley water through it and then create beer through your tap.
Yeah, like those like on tap Brita filters, right? But this is like
Yeah.
on tap kombucha.
Some people like kombucha, I guess. They must do, right?
Yep. All
Oh.
right, that's a short one. Just like an interesting technological idea. Up next, we have resolving the structure of phage-bacteria interactions in the context of natural diversity.
Yeah, this is an interesting one. So it's a population like look at a phage and different phages and their bacteria. So these guys went out
Mm-hmm.
into like into the sea and like pulled out a bunch of Vibrio bacteria and then sequenced them and all their phages that infect them. I think they did this thing called like
Mm-hmm.
a bait trap where they'd have a bunch of these and have a bunch of these bacteria in water and wait for the phages to infect them, then pull them out. And
Yep.
using some like really interesting genomics, they'd try to figure out the relationships between them. So looking at kind of the types of phages that infect certain bacteria, and
Yep.
also whether like one phage can like infect lots of them,
Mm-hmm.
and also looking
Yeah,
at
this is
like
kind of
the
like they wanted to learn something sort of abstractly about the ecology, right? Like we know that there's tons of phages in the ocean. We know there's tons of
mm,
bacteria, but like what about the host specificity? Like they want to find like a feature, right? They speak about like an older data set that was collected. This is like not easy work to do, right?
mm,
Because they have to grow a bunch of different bacteria and they have to treat them with a bunch of different phages to get a sense of like what those pairwise interactions are.
mm.
So like I guess they were talking about like a data set that existed is like one of the only data sets they have to work with in terms of pairwise interactions, bacteria and phages. So here's like the second time that someone's made a data set that they can start doing this type of analysis on.
Yeah, and they analyze the structures of phage interactions, and what they find is that there are like a couple of like phages that have a wide like variety of bacteria they can infect, but the
Mhm.
majority of them. So there's this graph that has like a line going through it, and the most of the interactions are basically one phage and one species of bacteria, and
Mhm.
that's it. But
Mhm.
there are a few exceptions to this. So there's let's see, there's there's some recently described phages called the Autolycoviridae, whose members
Uh-huh.
can infect some strains of like 6 species, and
Mhm.
they so each of these like, big boxes are like strains that are broad spectrum,
Yeah.
uh, but even if they yeah, so
Yeah. I mean, it's kind of there's just so many things out
mm,
there, right? Like it's impressive to be able to bring together experimental data to then shed some light about like what that interaction is like. They mostly focus, so because there's so much stuff to look at, they focus on like one particular type of bacteria here mostly, right? They're looking at Vibrio, and they also resolve them at a really high. They resolve them with not 16S. They resolve them with heat shock protein. So like they're building their phylogeny because like we know that they're basically all the same. Like they use like a bigger gene to try to find the relatedness between all of these different bacteria. Essentially like the tool that they're using to learn something about how phages and bacteria work out there in the world is they have this like big group of tightly related bacteria that are different, but they also are they're also like related in a certain way. And then they use that as like the way to, yeah, as you said, like a bait, like as a way of like polling the environment to see what sort of things kill them and at sort of what specificity. Like, are they killing everything in this family or just like a specific version? Because again, this is like, there's so many. The pre-existing hypothesis was that there's lots of one-to-one interactions.
Yeah, and what else was I going? Oh yeah, I was going to say that the interesting thing they find is that a lot of like so that sometimes what they saw was that like there is some gene like kind of transition, but not usually for between species that are killed by different bacteria or like if if there's a Vibrio species that is killed by two different like phages, you're not as likely to see a transfer of genetics. And what they kind of pare this down to is actually there's a lot of silent infections that happen. So the lysogenic phage. So when a phage infects a bacteria and then doesn't kill it, and then you have another bacteria infect, and not bacteria, another phage infect, and that's when a crossover can happen. So that's something that they could only really get from this data set because they had.
Sorry, so something infects a bacteria and then they get infected by another phage, and then that's what kills the bacteria ultimately? Is that what you're saying?
No, that's where the recombination, that's where the transfer of genes can happen. So that's when
I
new
see.
phages are born.
I see. This is like
So
a mechanism for creating diversity in phages.
yeah, so it's trying to analyze that aspect of like the phage life cycle and how they evolve.
Yeah, yeah.
So I found that quite interesting.
Definitely.
Because
That's crazy because again, then these phages go out in the world and they're pieces of DNA, right? Maybe some phage, but also some bacterial DNA, right? Like this is the complicated web of like gene transfer. It speaks to a little bit of like how we might hypothesize gene transfer works. It's not like specifically looking at that, but it's adjacent to that information.
yeah, so this is how phages can swap genes, and also how they can swap genes with the bacteria. There's a lot of and it's interesting for like the context of say looking at say phage therapy, where we have lots of trying to figure out like how phages evolve during infecting the bacteria and how they live in their natural habitat.
Sure.
And that's
Well, I mean, in the
really
context of phage therapy, I would say that we don't. Well, I mean, I don't know, but I would assume, I don't think we have phages just like floating in our blood. I'm not sure.
no.
Yeah, we don't.
I don't think we do. We do. I don't think we. I think we might have some in the gut. I don't know. Don't quote me on that. But
no, definitely in the gut. Definitely phages in the gut.
Yeah.
Yeah, but phages in the blood. But I mean, phages in the gut. Like if we're taking oral phage therapy, right? Like then this definitely would apply. But if we're doing like injected phage therapy, I don't know if people do that. If is that the way people talk about phage therapy?
I don't. Yeah, I don't
for
really
cholera, that's
know.
how it was. Oh no, for cholera it was ingested. Like that's the early DHRL stuff. Yeah, I actually don't remember now. We've covered a phage therapy thing. I don't remember how they gave it. Oh, they just did it. Oh no, that was just evolution in a flask. So this is quite different than that.
Yeah, so I mean, it's interesting getting this like population-wide like view on like how these like bits of DNA are spreading around.
Totally. Yeah. And again, like it's almost like trying to inform a theory. I think it is. It's trying to inform a theory about like population dynamics in nature, right? Like this is like in some ways, because they're using water Vibrio, right? It's specific for Vibrio, it's specific for this, and they hope that it'll be generalizable to thinking about this like water systems or bacteria and phage in general.
And I think another thing I wanted to bring up was that the importance of like phages that don't kill their hosts, because quite a lot of like studies like this are based on like plaque assays, which rely on like the hosts dying, and so that means that I can tend to like kind of limit our viewpoint somewhat to just the phages that kill the hosts, but
Yeah.
the phages that don't kill the hosts can also be quite important, and they're much harder to track down. And this study allows that, I think. So I think that's quite interesting.
Yeah. Okay. And moving on to our
Final
last paper. Yes. Massively parallel
massively.
Oh, go ahead.
Oh, no, you go.
Oh, yeah. Massively parallel characterization of engineered transcript isoforms using direct RNA sequencing.
Yeah, so so when like an RNA polymerase reads like off of like a DNA like template, there's stop codons which stop it from working. And these are usually characterized by like hairpins or multiple U like tracts, which can cause the RNA polymerase to slip off. And so I
Or
think
get
there's in this,
blocked, right? They can block
yeah.
the movement, and then because they block for so long, then they pop off. It's actually very, this is like if people go back to the how does remdesivir work, right? It's actually very similar thought process, right? Like introducing stalls, right, can force something to pop off after a while.
Yeah, totally. And so imagine you're like a synthetic biologist and you're trying to create artificial stop codons, and you find that some of them don't work, some of them are quite leaky. In this paper, they're basically taking these leaky stop codons and deciding, well, actually, that can be quite useful in of itself, because
Yes.
having something that has a percentage of going one way or the other can actually help us regulate certain like transcription.
Yeah,
So they
I mean,
create this
there's
thing that they call
Oh, I was gonna say like that. Yeah, that's totally that makes so much sense. Like because sometimes to control like the metabolic flux, right? You don't want to have a buildup of one product before another, right? Or like some toxic thing, right? Like you want to always be able to remove all of that. So you need more of the protein that does a critical step. It's like people need control over these things in synthetic biology. And yeah, it's interesting to think of faulty stop codons as a potential source of that control.
Yeah, so they create from these faulty stop codons something they call a transcription valve, something that will like turn off or on depending on how well this stop codon will react to things. So
Yep.
this paper is a lot of them experimenting with different ways of making stop codons, and then looking at the sequence using like the poly U to get things to drop off, and also looking at the structure of the sequence before as well to try and like modulate how well the stop or start happens.
Yeah, I think they're also this is like a design screening sort of experimental method where they say let's make a whole bunch of them and then characterize them, right? So like they don't have to know the design principles de novo. They can just sort of brute force it by creating all these different variants and then seeing how they behave. And I think what's critical about this paper is that it's reliant on this sequencing technique, the nanopore sequencing technique. Like you wouldn't get, I think it'd be very difficult to collect, to do this sort of research development style approach if you didn't have a nanopore, because the idea is that you'll be able to see how frequently, right, or you can actually see how frequently certain sequences get through the pore, and that's a rough readout on how difficult it was to string through that construct, and how much it's blocking essentially or partially blocking. Right, you have that data.
Yeah.
Yeah.
So there's a lot of that kind of testing and iterating that we are used to with bioengineering papers.
So what's interesting to me though is that they're trying to find things that work for RNA polymerase, but they're essentially testing using whatever pore molecule is inside of the nanopore. So I mean, I guess I didn't get a sense of that generalizability, right? Like, are they saying that the pore? I guess they're saying that the pore is pretty good because later they test it with, they test them in six. Oh yeah, in six they test it in like a system, right? They make a system where they want to add these valves into, and they find that it works, and they found those valves not by testing RNA polymerase, but by testing the ability of those things to go through the sequencing pore. So, right, that's quite fascinating to know that, like, I mean, it all goes back to physics, right? In some ways, it doesn't matter, like, it doesn't matter you're not using the exact thing. The fact that you're constraining that funny loop to go through, is enough. Okay, but another thing that this doesn't deal with in terms of design of them is, I also know that some terminators do interactions. Like the reason they terminate is because they interact with the protein itself. So like I'm sure that that's something that gets confounded in this design process. But maybe the idea is that they just need to discover a whole bunch of them, and then you never have to do this again, right? You kind of converge on your favorite valves, and those valves become like the de facto tools of doing this work.
Yeah, I mean, it's reminded by like one of the SARS-CoV-2 polymerases that like proteins that attach onto ribosomes to try and like regulate what goes in and out, and only make sure that only SARS-CoV-2. It's kind of like, it reminds me of that, but that's like almost the opposite, where they've got like a start codon that only works with this kind of lock that SARS-CoV-2 puts on the ribosome.
Yeah. But it's related, right? Because like this is in some ways like that, right? Like this is how learning about the way things actually work to make life go, like kind of feeds back in many ways into then how do we design stuff that we want to happen, right? Because like you need an understanding of how things work, or like the solutions that evolution found to help inform the way that we design things when we want our synthetic objects to work in a certain way.
Yeah. So I think that brings us to the end of our show.
Agreed. Yeah. Join us next week for more cool microbiology news.
And thank you very much for joining us. We want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. So science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
Yep, I totally agree. You can reach out to us over our Twitter at @MicroTWJC. We both believe that peer review is a process that we can all participate in, and so that we hope you had a good time listening to us ramble on about microbiology today. If you found something unclear, or just let us know in the comments. And we have that poll that's also in the doobly-doo below, right? That's going to close probably on Thursday, I think. So please vote so that we know what to cover in our next deep dive.
Yeah, it's been a pleasure chatting with you, Danny.
Same here, Fuzz.
Well, see you next week.